Showing here. I'm a consultant for a number of companies that are in the gene therapy space, both in hemophilia and outside of hemophilia as well. Well, what's the overall goal of gene therapy? It's to replace the dysfunctional gene in a person with an exogenous functional gene in order to cure the disease. That's the ultimate goal. For hemophilia, we have a moving bar. What we had as optimal therapies 10 years ago are very different than what we have as optimal therapies today. So the bar is going up, and as a result, the bar for gene therapy efficacy also needs to go up. And we need to be thinking in terms of a cure, not necessarily as a treatment, because we've got some good treatments. That, of course, is individual, and it comes down to benefit-risk considerations for each individual, as well as within populations of individuals. I think we're all aware of what gene therapy is. It involves the delivery of a new gene into the body, specifically for hemophilia, into the liver, into hepatocytes. And that's been accomplished with adeno-associated virus. Why do we use adeno-associated virus? Because it works. Our AAV infects us throughout life. There are many different serotypes of AAV that can infect us. It works with helper viruses. We do develop an immune response to it with antibody titers. It does not cause disease, fortunately, but those antibody titers can last for a long time and are cross-reactive among the serotypes because of the high homology between the serotypes. The AAV itself is comprised of two genes, a replication gene and a capsid gene. Those are removed, and then an optimized cassette for the transgene of interest, in our case factor VIII or factor IX, is put into the AAV, and then it's made by GMP means. These pre-existing neutralizing antibodies that are present in some individuals will prevent delivery of the transgene to the target organ, so that is a problem. Post-delivery, because we're administering hundreds of trillions to quadrillions of vector particles, we make very high antibody titers. And so because of the cross-reactivity among the serotypes, there likely is no redosing paradigm to be developed, which is a problem. It's a once-and-done kind of delivery. We also see a little mild toxicity with transaminase elevations that are due at least in part to a cytotoxic T-cell response against AAV capsid peptides. That also means that we see hepatocyte death as a function of the transaminase elevations and a loss of transgene unless we treat with immunosuppressive agents such as prednisone. AAV factor VIII, in addition, may also damage hepatocytes in non-cytotoxic T-cell ways that are not well understood and may involve the actual creation and synthesis of the factor VIII protein, which may have some intrinsic toxicity to the cell. And then finally, AAV has been considered non-integrating, but it does integrate. And when we administer hundreds of trillions to quadrillions of vector particles, the integration translates into millions of integrations per liver. No risks have been identified to date, but this is something that is a matter of long-term surveillance. I have been very interested in understanding and encouraging the field to understand more about the biology of AAV. What don't we know about it? What should we be expecting? How can we manage the results that are coming out that I'll describe in this talk? And as a result, we have a number of questions about the biology that are not keeping pace with the clinical trial developments. What are those clinical trial developments? Well, for hemophilia B, we're looking at gene therapy trials from several different places. The first trials used native factor IX, and this was the St. Jude UCL trial with Nathwani, Tuddenham, and Davidoff. And they achieved an average of 5% and had a dramatic effect on factor IX usage and annual bleeding rate as well. Nice thing about this is that it looks like it's stable out to at least 8 years, and so that's encouraging in and of itself. The Unicure trial used the same approach, same vector essentially, and found the same results. But then Padua came along. This is a hyperactive mutant that has a 5 to 7-fold increase in specific activity, and as a result, it can provide for higher levels at the same dose of vector. And that's shown here with the Spark Pfizer trial, where they're at about 20% factor 9. It looks like that's been stable for 3 years. And then a Unicure trial, which I'll describe, that's at about 40%. Key things to remember here, though, are that the inter-individual variability and the intra-individual variability are high. Durability looks like it's established for factor 9, and steroids have been required in a number of these individuals to prevent factor 9 loss when this cytotoxic T cell response arises. This shows you a little bit of the variability that I was describing. This is for fidanacogene elaparvovec, and this is the Spark Pfizer AAV factor 9 drug. And so over a 4-year period, we can see an average of 20% factor 9 activity, but we also see high variability. Here we have a mean of 26%, standard deviation of 13.2%. So this variability really means it's not fully predictable as to how good of a response a patient will get. And that has become apparent in a number of these trials. We also have a trial with from Unicure, etranacogene dezaparvovec, that is late breaking at this year's ASH meeting. They were able to show in their phase 3 trial at 6 months a 37% factor 9 activity, but again, I'll call your attention to about a 20% standard deviation and a range of 1 to 97%. So very hard to predict the response one will get with the gene therapy. What about factor 8? Well, the inter-subject variability is even a bit higher here than I think we've seen for factor 9, and there is still intra-subject day-to-day variability within an individual. With the trial that's gone out the farthest, the BioMarin trial, we see a lack of durability that I'll show you on the next slide. And the other trials are done in very small numbers of patients, don't necessarily show a dose-response curve. And the transaminase elevations have been managed for better or worse by steroids, but not necessarily as clear and clean-cut as they have been for factor 9, implying that there are other toxicities associated with AAV factor 8 that are not necessarily fully responsive to corticosteroid treatment. This shows the BioMarin trial, the lactocogene roxaparvovec, and what it shows is a 4-year frame looking at the amount of factor 8 by chromogenic assay. And so we can see at the end of year 1 in the high dose group, which is in blue, we have 64% factor 8 activity. That drops to 36% at the end of year 2, 33% at the end of year 3, 24% at the end of year 4. And the same applies to a lower dose of the AAV factor 8, which goes at the end of year 1 from 21% down to 10% at the end of year 3. So lots of inter-subject variability as well, as you can see, ranging from up to 180 units per deciliter down to close to 0 units per deciliter. And so we continue to see this variability in the factor 8 trials as well. On the other hand, despite the variability, it works. It decreases the annualized bleeding rate. It decreases dramatically the amount of factor 8 infusions that are required. This shows before receiving the vector, and then this shows after receiving the vector. But this is really what we would expect. And so while the various companies are saying it's a remarkable 91% drop in bleeding events, it's not really remarkable because at levels above 5%, it's exactly what we would expect. And it's exactly what Nathwani showed in his average 5% levels for factor 9. So this is expected. But the decrease that we see, the lack of durability, is unexpected, and that's potentially a problem as the years go on. In addition, because a number of patients have not achieved normal levels or near normal levels, factor supplementation is required for surgery as well as for some traumatic bleeding episodes. This is looking at a different AAV factor VIII from Sangamo Pfizer, giroctocogene fitelparvovec, and it is a phase 1/2 trial that goes out to about a year. I just want to highlight the fact that you can see the interquartile variability, the full variability here in the 5 patients that were treated. We have an average of about 71% factor VIII activity through a year, but it's not clear whether or not this will be sustained, and it may be that we see some decrease at the end of the year that we'll need to clarify as we look at the data from the next year. And so in this case, we also see a lot of variability. Well, what causes the variability? There's lots and lots of steps along the way, from the manufacture of the vector itself and the cassette, the transgene, to the infusion of the vector and the pathway that the vector takes from the circulation to the hepatocyte, internalized into the cytoplasm, moving into the nucleus, forming a second DNA strand, forming a stable episome, and then being able to make messenger RNA in order to make the factor VIII protein. That is a very individual series of events that occur in each person who receives the AAV factor VIII, and certainly has to account for a significant amount of the variability. Well, despite the variability, we have had some major advances over these past 10 years. This started with the Nathwani-Tuddenham-Davidoff trial that I've described, giving us an average of 5% factor IX. We were all very happy with that at the time. But now we see that with the patoimmutants, we can achieve higher levels of factor IX. It's not clear a 20% level is sufficient from the Spark Pfizer study, and they are planning a study using a higher dose of the AAV factor IX to see if they can achieve a higher level of factor IX, especially in view of the fact that Unicure and CSL, which is partnered with Unicure, have factor IX in 54 phase 3 subjects. That is about 37%. So lots of developments on the factor IX front. On the factor VIII front, we had a great surprise in 2016 with BioMarin, 7 participants that I showed you the data from. Using at that time the OS assay, the one-stage assay, we had 100% mean factor VIII at 1 year, which translates into about 64% by the chromogenic substrate assay. So this was exciting, despite the variability. But now we're 4 years in, and we see that 75% of that factor VIII's been lost. No change necessarily yet for the patients who are maintaining factor VIII sufficient to prevent spontaneous bleeding, but time will tell as we monitor that trial. In addition, we've got at the ISTH, Spark showed 1 year data on their factor VIII study in 12 participants, no dose response. 2 of them lost all of their factor VIII, presumably due to a cytotoxic T cell response, and there's high variability among the remainder. And then also recently, Sangamo Pfizer showed 5 participants, as I showed, some achieving normal levels of factor VIII, and we'll have to see whether or not this remains durable. So the surprise happened last August when BioMarin, which had submitted preliminary phase 3 data for an accelerated approval with the FDA, had the FDA reject their drug. And the FDA said that the results from the preliminary phase 3 trial data did not look like the results from the phase 1/2 trial that I've shown you. FDA also said that the durability is a question, and so they've asked for 2-year data from BioMarin before submission for approval of the full 134 patient phase 3 study. So as we look at all of the variability, we can ask ourselves biological questions. What we know and what we don't know today. What we know are the known knowns. These are the questions we know we can ask, and we know that we've got some answers to. We know that some patients can get greater than 40% factor VIII and factor IX and do quite well with the AAV gene therapy, have largely protective levels. And the remainder of the patients, most of them, have some degree of protection. May not be enough for surgery, but may be enough for spontaneous bleeds and many traumatic bleeds. We still have major questions about the variability and the predictability, and the fact that there are a few patients that just don't get a response, and we don't know why. And that durability for factor VIII remains an open question, although it looks like we've got good durability with factor IX from multiple clinical trials. So then we get to the more perplexing part, what we don't know. And these are the known unknowns. So these are the questions that we know to ask, but we don't have answers to. And then the unknown unknowns, the questions we don't even know to ask. And this variability and predictability that we have trouble with is a major question that we don't have answers to. We know that each individual is variable and that must be contributing because we're not in 20,000 liter stainless steel tanks making recombinant protein. We're in our human bodies trying to do the same thing. What's the range of host immune responses to the virus? I don't think we understand that. We don't understand the innate immune responses, the full range of the adaptive immune responses. We don't understand whether or not there are other causes of liver damage, especially in the case of factor VIII. And we don't understand the potential risk of integration in the patients. So we're left now with evolving perspectives as we look at the data and we match that up with the technologies that are furthering hemophilia treatment in the protein replacement arena. Our perspectives are changing. We also know that it takes a long time to develop a drug, and the gene therapy development pathways probably take more than a decade to go from start to actual product approval. And so a lot can happen in that period of time. Access to extended half-life factor IX and emicizumab, those two drugs have significantly changed the market for hemophilia and have elevated the game for gene therapy. What was exciting in 2011, 5% level, is not as exciting today. And the idea of curative therapies getting into the subnormal to normal range may be required to compete with these other technologies. And the EHL factor IX and emicizumab aren't the only other technologies. There are others that are in clinical development that look like they will also improve the treatment options and decrease the burden of therapy, as well as better efficacy for patients with hemophilia. So our most recent expectations for gene therapy don't necessarily match the results that we're seeing in all of these clinical trials. On the other hand, and this is an especially important caveat, we also recognize 70% of the world have minimal access to therapies. And if one can achieve stable 5% levels in patients, that would make a world of difference. And so we've got a dichotomy here between individuals who have access to these newer therapies and individuals who don't, with regard to the benefit risk of gene therapy. And I'll leave this slide with a quote from Peter Marks, who's the head of the FDA Center for Biologics Evaluation and Review, the group that reviews gene therapy. And what he said at our recent gene therapy roundtable at the WFH was that hemophilia gene therapy must be taken in the context of available therapies. And we have a low tolerance for adverse effects. And so those are sobering thoughts. I'll leave you with an open series of questions here. These Generation 1 AAV therapies are likely to get approved in the next 2 to 4 years by regulators, despite the variability and unpredictability of responses and the question of durability for factor VIII. For AAV, we need to understand the biology from start to finish, and we're not doing a good job of that. The companies for the most part, are not addressing this. They're just breezing through, moving forward on phase 3 clinical trials without stopping to ask important questions about the biology that I've described in this talk. I also want to point out that AAV isn't necessarily the only answer. There are AAV alternatives that are moving toward or entering clinical trials. That includes lentiviral vectors, non-viral gene therapy, and cell-based therapy. And so as these move forward, we'll determine their efficacy and safety parameters, and they may offer some competition for AAV. And what I might leave you with in terms of the price of entry, I do think that the biology is an important point for the price of entry, and a broader issue is the ability to redose. If we can't predict the response, if we don't know if a patient's going to get a poor response or a high response, then it may be the ability to redose, which can come with some of these AAV alternatives, might become the price of entry for gene therapy in hemophilia. I have no doubt gene therapy will make a major impact on the management of human disease, including hemophilia. The first question is when, and the second question is how do we accelerate this? With that, I thank you for your attention.